Good evening, everyone. Thank you for coming. It gives me great pleasure to invite Professor Tom Seeley from Cornell University, who has spoken here in person in the past, now speaking by Zoom, and he's going to talk to us about nature-based beekeeping. So, Tom Seeley.
Thank you. It's my pleasure to be back, at least virtually. And I'm sorry that I wasn't able to make it over the last time, which was at the very start of the COVID epidemic. But great to be back. As you can see, I've juxtaposed two individuals here, and I'd like to see if you know who they are. Think about that for a second. Brenda Langstroth and Charles Robert Darwin. And you might be wondering why I'm juxtaposing them. It's not because they share some features, though they do share some features of their lots of hair on their heads. Birth dates, were born almost at the same year, lived almost the same lifespan. But what really makes them interesting or important today is that they both had a fascination with honeybees. With Langstroth, of course, it was the invention of the movable frame hive. He recognized the bee space and figured out a way to make use of that. For Darwin, it was also comb building. He was a admirer of comb building. In fact, in his book On the Origin of Species, he writes a chapter on behavior. He describes comb building by the bees as the most wonderful of all known instincts. And both of these gentlemen have made advances that help us with our beekeeping. Langstroth, of course, it was the concept of the movable frame hive. Darwin, it's the concept of evolution by natural selection. This point, just probably everybody here knows what that means, evolution by natural selection. It means survival. Individuals within a population vary in some traits, and those individuals that have traits that give them better survival and reproduction, those are the traits that get passed on. And over time, the population's composition comes to have the traits that are helpful to the animals or the plants. So how does this concept of natural selection help us with our beekeeping? Well, it tells us that everything that honeybee colonies do when they're living in nature is done to favor their survival and reproduction. When I say everything, I mean everything. And I've just shown a few pictures at the bottom here that illustrate the amount of brood that the colony rears, the amount of pollen it collects, the amount of honey it stores, even the amount of brood food that's put down in the cells with the young larvae. That also is an adaptive trait of the bees. So knowing that everything bees do when they're living in nature helps the bees, tells us that it's practically pretty important to look at how bees live in nature. And that's what I'm going to be talking about today, what I'd like to call nature-based beekeeping. And as we'll see, it's a set of practices that enable honeybee colonies to live as much as possible. It's hard to do it, make it completely natural, but as much as possible as they do in nature. And the logic here is that this enables colonies to use their survival skills, to use them fully. And it turns out it can also be a low-cost way of beekeeping. Now, a caveat up front, or here, nature-based beekeeping is certainly not for everyone. It's not for large-scale beekeepers, and it's probably not for urban beekeepers, because in urban settings, there are problems with swarms. But it is an option for a small-scale beekeeper, not a small-scale beekeeper who wants to produce a miticide-free honey and is satisfied with modest honey crop, something around 10 to 15 kilograms per hive, and who wants to let his or her colonies live naturally. And that's me. That's a description of me. Small-scale beekeeper, miticide-free honey, modest honey crop, want my colonies to live naturally. Maybe for some of you too, hope so. But you're probably wondering, hey, Tom, is it wise to allow colonies to live naturally? Don't honeybee colonies need our help to deal with varroa? Well, that is what I believed firmly before 2002. I thought that honeybee colonies, now that varroa is here, they do need our help. And back then, I was treating my colonies with fluvalinate and thymol. But in 2002, I started noticing that I was studying the colonies in the forests around my home, and I discovered that they were still thriving. I did a survey of the colonies in 2002. The wild colonies were still here, even though Varroa had arrived here in our hives in 1994. So I thought, wow, what's going on here? What's going on out in the woods? Now, let me explain where I live. This will help you get oriented to the context of my beekeeping. I live in New York State, up here in the northeast part of the United States. It's a rougher climate than yours. We get very cold winters and very hot summers. And the place where I was watching and monitoring wild colonies of honeybees was in this forest called the Arnott Forest. It's owned by Cornell University. And the reason this forest turns out to be very important is it's the one place in North America with data on wild colony abundance before and after the arrival of Varroa. Varroa showed up in my hives. I first saw them in 1994. I had studied the wild colonies and measured their abundance in this forest before and after the arrival of Varroa. And this slide illustrates my first survey of the wild colonies in the Arnott Forest. It's outlined in the black heavy line here. I've forgotten the volume or the area of the forest. I think it's close to 20 square kilometers, maybe 15. Anyhow, in 1978, I went out and learned how to go bee hunting, or beelining as it's often called. And I didn't make a comprehensive survey of the forest, but I did beeline a lot of bee trees in this forest. One here, 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 here. I mostly worked in the south part of the forest, but I didn't get this section and I didn't get this section. But I got an estimate of the colony density, assuming I had surveyed half the forest, and I found 9 colonies. It looked like there were about 18 colonies in the forest. So there's about 1 colony per square kilometer. And it was also revealing to me to know that the nearest neighbor distance for the colonies that I found was 580 meters, far apart. And that's relevant to what we'll be talking about. And if you're curious about how you go beelining, go bee hunting, find wild colonies, and you can do this in England or anywhere in the UK, I was so entranced by this process of bee hunting, I wrote a book about it, Following the Wild Bees. And this is a typical nest entrance of one of the wild colonies that I was finding in the Arnott Forest. It's a small entrance, you can see. And as we'll see, this entrance is high up the ground. You know, as I was trying to say earlier, when I went, I had in 1978, 1 colony per square kilometer. Varroa came in 1994. And in 2002, I went back to the forest, the Arnott Forest, and I surveyed the colonies again, and I found 1 wild colony per square kilometer. And I had expected the colonies to be all wiped out, but no, they were still there. And this raised the question, how is this possible? Is it the case that these colonies are so remote that they didn't have Varroa? There aren't any beekeepers in the area, but so maybe they didn't have Varroa. And I should explain, this is what the landscape of this Arnott Forest looks like. And it's the landscape that is to the south of Ithaca, New York. Up north of Ithaca, it's flat, excellent farmland, but south of Ithaca, it's very hilly. It was long ago abandoned by Irish immigrants that tried to settle on it, stayed for a generation or two, and then abandoned it, and it's grown back to forests. So all I know is that there were still wild colonies, as many wild colonies in 2002 as in 1978. But I didn't know whether these wild colonies were living with Varroa or not. So what I had to do was I put up bait hives in the forest, in the Arnott Forest. And over the years, summers of 2003, 2004, I caught 11 swarms in the forest. And once they're in my bait hives and they're moved into movable frame equipment, I could check them for Varroa. I could measure the Varroa loads in the colonies. Every colony had Varroa, but the Varroa counts were at very low levels. I can't remember the exact numbers. It might have been 1 or 2 per 300 bees, something like that. I think that's what it was, in fact. And so I drew the obvious conclusion that the wild colonies in the Arnott Forest are surviving with Varroa. Somehow they're surviving with Varroa. And that sparked a whole line of investigation, which I don't have time to go through today because it's a little off topic, but we did a very sophisticated genetic analysis looking across the entire genome of the old bees I had collected in 1978 and stored in the Cornell insect collection, and compared them to the bees I was collecting post-Varroa. And there are huge changes genetically in the bees between those two types. There were many, many for example, there were many matrolines or queen lines that were wiped out, but there were changes throughout their genomes. So this raises the question, how were these wild colonies in the Arnott Forest and elsewhere in the woods around Ithaca, how were these wild colonies controlling Varroa? Well, it's like every question in biology. There's a genetic aspect and there's an environmental or lifestyle aspect. So I'm giving you a summary of the answers to the question, good genes? The answer is yes. And so we'll see, they're good at killing by biting the adult Varroa. And these wild colony bees are maybe also good at uncapping and recapping brood cells infested with Varroa. I haven't measured that sufficiently precisely to be sure, but I do see that they do that. But it's not just good genes, it's a matter of good lifestyle too. And we'll be looking at this. And when I say lifestyle, I mean things like colonies swarming every summer. This creates a brood break. And when there's a brood break, the Varroa can't hide, and thus they're exposed to the adult bees, the adult who can damage them and throw them out. The another thing about lifestyle, and this is just one more, but there's a bunch, the nests in the wild are small. They're about the size of what would fit in one deep 10-frame Langstroth hive body. And this means there's rather little brood for Varroa reproduction. And because the colonies, especially in late summer, the colonies are filling up most of the combs in their nests with honey. And so the brood nests get small quite quickly early in the summer season. So now what I want to do is compare in detail the way in which wild colonies live and the way in which managed colonies live. And because the logic here is that in nature-based beekeeping, the goal is to enable managed colonies insofar as possible to live their natural way of life. So let's compare them, the way their lives work. It's going to be an 8-point comparison between the two types of colonies. First point of comparison is for the wild colonies, they are genetically adapted to their location. The managed way of life, at least over here in the United States, is often the colonies are not genetically adapted to their location. Let me give you an example of why this is. There are a lot of queens shipped around in North America. And here's an advert in one of the bee magazines, which I think is hilarious. The ultimate queen bee from Hawaii. Well, they're selling these ultimate queen bees from Hawaii all over North America and Canada, Mexico, all around. And of course, these might be just dandy queens for Hawaii, but they're not necessarily going to be well adapted to all of the locations to which they're being sent. But another way, another difference between the wild way and the managed way of life. Colonies in the wild live widely spaced in the woods. I've already mentioned that in that Arnott Forest, the nearest neighbor distance is on the order of more than 500 meters apart. Well, here's one of my old bee yards. Colonies, it illustrates what is a common picture, not for me these days, but back in the day, this is how I kept my colonies crowded together in an apiary. Third comparison between wild way and managed way. In the wild, colonies live in small nest cavities, about the volume of one deep hive body. Here's a distribution. Early in my career, I collected a lot of natural nests of honeybees. And one of the things I did is I measured the volumes of the cavities these wild colonies were living in. And here's the distribution in liters down here, but maybe a more useful set of units or volume units is number of deep 10-frame Langstroth hive bodies. So most of the wild colonies are living in cavities that are a bit smaller or a bit larger than a single deep 10-frame Langstroth hive body. In contrast, the managed way, well, this is what a reader of a bee supply company in the United States, North America, sees is to be considered the best and most complete wooden kit. And whopper, it's a biggie. It's got 2 deeps, and I think it's got 1, 2, 3 mediums for honey supers. Now that is approximately 4 times bigger than what a colony would be living in the wild. Much more brood, it's sort of a bonanza for Varroa. And these colonies tend not to swarm. We'll come back to that. Another comparison between the wild way and the managed way. In the wild, the tree cavity walls are thick for good insulation, 10 plus centimeters, and they're coated with propolis. Here's an example of just one little portion of a wall. It's actually at the entrance of a nest. Managed way, well, the commercial hives walls are thin, about 2 centimeters, and the interiors are not coated with propolis because the wood is planed down so smooth, and the bees aren't stimulated, don't sense the cracks and crannies where they want to put the propolis because they can't keep those areas clean. But in hives made of planed lumber, it's not so hard for them. Fifth comparison. In the wild, colonies build drone comb freely, and they produce lots of drones. When I studied the nests of wild colonies, one of the things I carefully measured was the percent of the comb, total comb area that was built of drone comb, and it was about 15%. It was remarkably consistent too. So those bees, they're very careful to allocate a good body of comb to making drones. And that, of course, makes perfect sense in terms of genetic success of a colony. Drones and queens are the two ways that colonies pass their genes on to the future. So they produce quite a few drones. But in the managed techniques that I was taught, colonies are discouraged from building drone comb. We give them plastic comb foundation so they can't tear it out. And so they're stuck building just gobs and gobs of worker cells, almost always worker cells. And colonies produce few drones. Another point of comparison, the height of the entrance off the ground. Out in the Arnott Forest and other places in the forest around Ithaca, the average nest entrance height is 8 meters. That's me. And I'm not quite 2 meters tall. So this was a big one, tall one up here, but that's not unusual. And the managed way, of course, we keep our hives down at ground level. That's handy for us. Big difference, though. Another difference is in the wild, colonies have diverse pollen sources because it's all wild. It's what's wildflowers, and there's all sorts of wildflowers growing in nature. The opposite extreme under a managed situation is the almond orchards in California, where for a while all there is almond nectar and pollen. So sometimes colonies in a managed settings have very non-diverse pollen sources. And an eighth point of comparison, in the wild, colonies are not treated for diseases. And no surprise, bees evolve resistance to Varroa. I'm sure that's happening in parts of the UK as well. But in the managed way, colonies are treated for diseases, including Varroa, so bees do not evolve resistance to Varroa. The selection pressure of Varroa is not being exerted if the Varroa is always being killed off by mite away quick strips or whatever. So those are the 8 points of comparison between the wild way of life and the managed way of life. And what I want to do now is go back through those 8 points, but now with respect to guidelines for nature-based beekeeping. So the outline for this nature-based beekeeping has 4 main components. The first one is, and it's a very important one, keep bees that are adapted to your location, that are locally adapted. And there's several ways you can accomplish this. You can rear queens from your best survivor colonies, which is what I like to do. Or you can capture swarms with bait hives out in remote places. I don't know if there are, I'm sure there are remote places in Wales and some parts of the UK where you could do this. You could put out bait hives and you'd be likely to get a swarm from a free living colony. I like to do that too. That's fun, and as we'll see. Or you can purchase queens from a breeder who produces locally adapted queens and ideally does not treat for Varroa. I don't know if those exist in the UK. I hope so. Certainly should be a market for that. My second guideline for nature-based beekeeping is to house your colonies in small hives. And when I say a small hive, I mean one deep 10-frame hive body plus a shallow super, maybe two, a shallow super over a queen excluder so you have a honey crop, get some honey for extraction or produce comb honey. And this is a guideline that is based on an experiment that I did where I compared the health and survival of colonies that were living in one deep hive body, and they were simulated, so I call them simulated wild colonies. They weren't perfectly simulated because they were close together, like in an apiary, And there were 12 of them. And then nearby, this is part of a storage shed, and we see another corner of that end of that storage shed over here. So nearby to these colonies, there were 12 more colonies which were managed for honey production. They were put in big hives. So this is the point of this we're looking at now, comparing the fate and health of colonies housed in small hives versus big hives, honey production hives. What did I find? Oh, and I want to stress here, these colonies were just installed um, uh, on new combs, empty combs. They were installed as what we call packages, 3 pounds of bees and a queen, and all the queens came from the same place. And none of the colonies received Varroa treatments. It was just sink or swim for these colonies. And what we then, I followed them. I'll just summarize what happened in terms of the percent of the colonies that were alive after 2 years of this, just leaving them alone and letting them go. 83% of the simulated wild colonies, 10 out of 12, were still alive and they were in good shape. 2 died, but only 2. Over here, in these honey production colonies, there were only 17% still alive, 2 out of the 12. And interestingly, you might be interested to know, well, there were 2 that survived over here, but boy, you wouldn't want to have those bees in your hives. They were very defensive. And I think they were defensive not just against beekeepers, but probably also very defensive against mites, though I didn't actually test that. So having colonies live in a simulated wild colony hive situation really helps the colonies. And, as for reasons that I alluded to already. These colonies living in these small hives produce swarms. They export mites. They have small brood breaks. They fill up their combs with relatively early in the summer, so that reduces the brood that's available for Varroa to reproduce. They have a lot going for them, except they don't make much honey. Here's my third guideline. And again, it's based on thinking about how colonies live in the wild. Space your colonies as widely as possible. And here again, I did an experiment uh, which allowed me to get some evidence that spacing the colonies is a big deal with respect to dealing with Varroa. So I set up 2 apiaries in the same general area. Here's a bird's eye view of the whole area. There was a group of these 12 colonies were assembled here in an apiary. And then nearby, there was an old radio astronomy telescope array over here. I spread out 12 colonies, but here they were not so close together. Here's a scale. They weren't very far apart. They were maybe 50 meters, 20 meters, more greater distances, somewhere in that range of 20 to 100 meters apart. So they were, I would have to say, I would describe them as moderately dispersed colonies. And again, there are 24 colonies here. None of them was treated for Varroa. And then I followed the colonies. And again, it was just sink or swim. I just let them go. And they were all in 2 deep hive bodies. So not quite like a natural situation, but that's how I did the experiment. What did I find? I found that after 2 years, 0 of the colonies over here were alive. 0. Didn't surprise me because I was watching the mite counts go up in them. Over here, there were 5 still alive after 2 years. And that did surprise me because these colonies weren't spaced out as widely as in the forest. So it seemed like there was much more potential for colonies to rob colonies here than in the woods. Certainly it wasn't the opportunity or for robbing wasn't as strong as in the apiary, but it seemed to me, in hindsight, it seemed to me that I was surprised to get 5 still alive over here. But those were the results. And as I say here, I regret setting up this experiment with the 2 groups only 200 meters apart. And this is another shortcoming of this experiment, which I'll confess, because there was much potential for robbing honey when these colonies were starting to crash, die out because of I wouldn't be surprised if bees came from some of these colonies over here, robbed them out, and brought home lots of mites. And probably that's why That's what doomed some of the colonies over here. I wish I had done this experiment differently with the two groups farther apart, and within this group, the colonies farther apart. But it gave us some information. And this robbing is something that we've looked at more closely, and it is really why all of these colonies died. Because I could see some of them were collapsing, but what I wasn't studying was the robbing of them. But I then did a study on robbing. And this is a complicated graph, but I'll walk you through it. In this experiment, we set up 3 colonies, and we call these the mite donor colonies. And these are the mite counts in our mite donor colonies. These are the phoretic mite counts, the ones that are on the bees. And in these mite donor colonies, you could see by end of August, this is the beginning of September, they were getting high mite counts, on average about 60 mites per 300 bees. So those colonies were about to collapse. Nearby, we put mite receiver colonies, MRCs, and those were 1 meter, 50 meters, and 300 meters from the little cluster of mite donor colonies. And here's the key thing that's shown in these graphs. The mite receiver colonies, they were doing fine until these colonies started to collapse. Their mite counts went sky high. They were dying. And as they were dying, the mite counts were going up in the other colonies. This is a mites per 300 bees. And here's mite drop counts with sticky boards. Both measures indicated that when these colonies were crashing and becoming vulnerable to robbing, the mite counts went up in the nearby colonies. And those nearby ones, some of them were very nearby, some were 50 meters, some were 300 meters. And even the 300-meter colonies, some of those were picking up mites, probably from robbing. So robbing's a big deal, and it's one of the real shortcomings of conventional beekeeping. It creates the opportunity for robbing and spreading of mites. And the beekeeper really has to keep on top of that. Okay, guideline 4 for nature-based beekeeping. Line your hives with propolis collection screens or build them of rough lumber, because you want to stimulate your colonies to build a propolis envelope. It functions as an antimicrobial shroud, and there's very good work from Marla Spivak at the University of Minnesota that shows that having this propolis envelope lowers a lot of the disease stress on the workers in colonies when they have that. This shows how she stimulated colonies to put down lots of propolis on the walls. She'd staple to the walls of some of her hives in her experiments these plastic sheets of propolis collection screen, and then when you take it off, you can seal all the propolis. It's not as good as the bees would do if the lumber were rough, but it's way better than not. My 5th guideline is that you provide your most resilient colonies, lowest mite count colonies, with 10 to 20% drone comb. And why is this? Well, the drones are half the story of a colony's genetic success. So you want to make sure that you're really good colonies, ones that can fend off, that keep having month in, month out low mite counts. You want those ones to be making lots of drones and spreading their genes in your area. You want to promote the genetic success of your best colonies. But that does mean you have to monitor the mite levels, because you don't want to have the ones that are lousy at dealing with mites making lots of drones. Just the good ones, the best colonies. One of the things I like to say when I show this photograph is I think this is a very charming photo of a drone. It bespeaks the nature of a drone, because even before he's clambered fully out of his cell, he's tongue out seeking some food. Drones are hungry guys. I love it. Okay, back to science. 6th guideline for nature-based beekeeping: keep the nest structure intact. And what I mean by this is put each frame back in its original location and with its original orientation. No reversing of brood boxes, no breaking up the brood nest, that kind of stuff. The bees work very, very hard to organize their nest. And a lot of the beekeeper interventions, often for the sake of swarm prevention or swarm control, is to mess up or to disrupt the bee's nest. I'm sure that's not healthy for the bees. You spread out, if you alternate combs with brood and combs that are empty, frames of empty comb, you're really putting a stress on your colonies. So guideline number 6 is to do as it in nature. Keep the nest structure intact. The bees are the best beekeepers. They know how their nest should be organized. Seventh guideline, and this is probably more pertinent in North America, where I am, where the winters are wicked. Provide only a small entrance at the hive bottom. I don't know about your bees, but my bees have bored top openings and close them with propolis because they know that they're going to lose a lot of heat at the top of the hive. And at least where I live, the bees depend on condensation inside the hive for their winter water supply. We have long cold winters and the bees can't go out and get water. So I found for my bees that they really seal up the top, any top little opening crack with propolis. So they're trying to keep the nest well sealed up and also so heat doesn't go out and so the water can condense inside the hive to give them water. The thirstiest bees I have ever seen in my laboratory were bees I had in an observation hive in my office. And it was a heated room. There was no condensation inside the hive. Bees got terribly thirsty. Some of them got so thirsty that they did these marathon waggle dances on a day when they could go out and get some snow melt. Now, one bee did more than 300 circuits of a waggle dance to advertise the availability of water. I have to say more than 300 because I didn't see her start her dance. So what I use for entrances, they're near the bottom of the hive, as you can see, and I make them the same size what I see in the wild, about 10 to 15 square centimeters. And I stick a wire mesh screen to keep them out of them. And here's my eighth guideline. And this is the one that is probably the hardest for people to swallow. It is refrain from giving colonies chemical treatments for varroa. Refrain from giving your colonies chemical treatments for varroa, because when you do that, you're preventing natural selection from working. If a colony needs to be treated for varroa, it's not a colony you want to keep. So what I do and what I recommend you do is you monitor the mite levels in your colonies. And if the mite level gets high, more than 5 to 10 mites per 100 bees, then euthanize your colony. You can do this a couple of ways. The one that I usually use is I just pour warm soapy water into the hive at dusk, having screened off the entrance. And the reason that you want to euthanize these colonies is, for one thing, it eliminates your non-resistant colonies. But equally important, it avoids producing, some beekeepers call them mite bombs, but that name doesn't work perfectly because really what you're creating is robber lures. And those robber lures, as I mentioned already, are the source for many colonies of suddenly having high numbers of varroa mites in them. So this is about being thoughtful about colonies nearby. And there is an alternative to euthanasia, but I'm not sure it works for very many people. It is to treat the colony with a miticide and then requeen it with a queen of resistant stock. And you could do that early in the summer. That could work well. But that depends on getting a queen of resistant stock. And mm, they do exist, but they're not easy to find in my experience. But maybe in Europe, it's easier for you. Now, you might be wondering, well, okay, Tom, but what are your success or survival rates of colonies using these methods? Well, let me tell you. Here it is. First of all, I'll show you that some of the colonies I keep in this nature-based beekeeping fashion. Here's a colony here tucked against an old barn. Here's another one out on the edge of a field, and here's another one again on the edge of a field. They're widely spaced. They're in a small hive. They have a small entrance opening. The hives are not well insulated. These are old ones. I'm switching over to hives with much better insulation now, and that will help make things even better. But there's just one entrance at the bottom, no top entrance, just one deep hive body. These had a screen on them because I wanted to do varroa counts, mite drop counts. These are the numbers that I obtained. Summer survival, I get from spring to fall, like October, 97% of the colonies are alive. Winter survival, it's 84%. So multiplying those two, you come up with overall for a year, I have 81% survival of my colonies that are not getting treated for varroa, not getting chemically treated for varroa. Of course, I'm treating them in other ways by giving them drone comb, small hive, and so forth. And what I do then, so I got to make up 20% of my colonies approximately each year. And I replaced them by catching swarms that I set in remote places, off in the hills, away from where I know there are no beekeepers. So I don't know that, of course, doesn't work for probably for most of you, or many, at least many of you. But for those of you that do live in remote parts of the UK, that is a good option that you have. And I know the work that's being done by coming out of Francis Ratnick's lab, where they've been looking in various locations, particularly in some forested areas. They're finding lots of wild colonies, both in southern England and middle regions. And I know there are other individuals looking around in England and Scotland and are also finding more colonies than you might imagine. Now, we've covered a lot of ground in this talk, and I don't know about you, but I really often I find it much more effective to read about a topic than just to hear a talk, though the talk can often get me going and on the topic. And so I wrote this book some years ago, I guess it's about 5 years ago, and it provides more information on how honeybees live in nature and how to pursue nature-based beekeeping. It wasn't written to be a manual of nature-based beekeeping, but it provides the biological basis for understanding the logic of nature-based beekeeping, the things I've been talking about. And I have one more slide, and I was asked to show this slide by the folks at Bees for Development, because I do have a new book about honeybee behavior. This is not about how colonies live in the wild. This book is all about 20 mysteries of honeybee behavior that I solved. And it's hot off the press. It was released 3 days ago, and you can purchase it online at beesfordevelopment.org, shop.beesfordevelopment.org. And some of those copies are signed. So that's a little promotion I've inserted for support of Bees for Development, which is an organization I think the world of. So I guess that's the end of my talk. We could do questions.
So the first question is, with your small hives, are they getting so full of honey that they don't have any room for brood, and therefore they get a brood break, which could improve their resistance to varroa?
Yes, I'm glad that question was asked. I tried to say that in the talk, but I said a lot of things. Yes, that's right. Come August, the brood nests in my colonies are quite small, and by September they've got that nest almost virtually full of honey and pollen. So yeah, there's an early shutdown of the brood rearing and thus of the mite production. That's an important part of this, of the way these colonies are surviving with varroa.
Do we have another question? So the question was, do you have to use this as a sort of integrated pattern? And if you were to just stop treating in isolation, is that likely to cause problems, particularly in an urban situation?
Oh yeah, this is not an approach that works in an urban setting. Simply because you've got so many other colonies around, and if they're not being treated or whatever, your colonies will, and if some of them die out, as we saw here, the robbing is a great way for mites to spread. Yeah, this is not going to work in an urban setting, I'm pretty sure.
So do we have another question, please? The question was, how do you set up your small colonies for efficient overwintering?
I don't do as well as I should, I'll say, first of all. The main thing that I do is I put 2 inches of foam on top of the inner cover. I put 2 inches of foam in between the inner cover and the outer cover, so there's not a lot of heat lost out of the top of the hive. And that's where I get to my new hives, where I'm building hives with 2 inches of good foam insulation on the walls. So I have the walls well insulated as well as the top. And that's the investigation that I'm doing right now, is the effect of that super insulation on colony survival over winter. And its help in the summer too. I don't know if you've ever reached into your hive where the sun is shining directly on the hive and you touch the inside of the wall of the hive. It's very, very warm. The heat is radiating off that. Goes right through that wood. It's nice for the bees in the winter, but probably is pretty tough on the bees in the summer. Big heat load into the nest.
We have a question right up at the back. So Chris's question is, did you harvest any honey from any of the hives in your comparison experiments?
Yeah, the dead outs, the colonies that were in those two-story hives that were crowded together, they had a lot of honey in them. So we did harvest honey from those. Yes, indeed.
So Martin's saying, do you always allow your colonies to produce a copious amount of drone brood, or you always allow them to do that?
Yeah, if it's whether it's an 8-frame hive or a 10-frame hive, I put in one frame, which I have equipped with a full frame of drone comb, because you can buy drone comb foundation. So when I set up my combs, I make about 10% of them with drone comb foundation. I wish I don't have a good way of giving the bees 15%, but so I give them 10%. And so because I'm only giving them 10% or 12.5% drone comb, they often squeeze in some more drone comb elsewhere. That's fine by me. I'm glad that question is asked, because when you provide a frame or frame or 2 frames, whatever size hive you're using of drone comb, you get a lot less burr comb built elsewhere in your hives. Burr comb that's drone comb.
At the front there. So do you inspect your hives, or do you just leave them to it?
Mostly I just leave them to it unless it's an experiment where I need to monitor things like the mite levels. If they're colonies I'm managing for honey production, I leave them to it. And that's certainly the case in the hives that I'm showing in this slide that you might see, where I was just testing their ability to survive. Yeah, those colonies were just left to go on their own. If I wanted to get some honey from a colony, you of course you can take out frames that are filled with honey, and in the middle of the summer, and the bees will have time to refill them before winter. So that is still a way to get a honey harvest from these colonies.
Right up at the back there. Do you use open mesh floors on your hives?
No, I haven't used those. It seems so unnatural. And I think I worry about it being very drafty and cold in the winter, so I don't use open mesh floors. I don't think there's much evidence that it helps with the mites either, but but to answer the question, no, I don't use open mesh floors. Again, it's unnatural, very unnatural.
Yeah. I suspect Tom's winters are considerably colder than many people in Britain, actually. So I should say, Tom, that open mesh floors are quite popular here, particularly in the sort of southern England. But as I say, I suspect your winters are considerably colder than ours are. So.
Yep.
Yeah, so the question's about urban beekeeping, and in urban beekeeping, it's quite popular to put hives on roofs. So, do you see the advantage in that the hives are away from the varroa mites and so on? I guess there is a sort of maximum desirable height. I know in London there are people who put hives on the top of sort of 10-story buildings and so on, which doesn't seem to be particularly good to me. What are your thoughts, Tom?
Boy, I don't have any well-informed thoughts on this, because don't have experience with an urban situation. What I can say though is that one thing, I didn't talk about this, but one thing that we have learned is that colony to colony mite transmission can occur when mites fall off, or somehow get off of a bee onto a flower. And these mites they know all about how to get on bees. And so they will climb right onto the next bee that comes to that flower. That said, I don't know whether having your hives well off the ground, so well away from where fallen mites are going to be, because some of those fallen mites, if they're falling off a bee, would be in a flower and thus could be brought home even if the colony is living high above ground level. Interesting question. Yeah, that's with almost everything with bees, we need to know more.
I mean, presumably in your hives in the forest, there are a choice of heights of cavities, and your bees are, preferentially choosing the ones that are sort of, 20 feet off the ground or so. So that there's obviously a sort of optimum height that they seem to prefer.
Yes, the reason I'm quite sure that the reason they prefer the high one, at least one reason they do, is because they're less noticeable by bears. Um, I didn't explain this, but when I put my bait hives up in the Arnott Forest, I got a quick lesson on just how savvy black bears are in recognizing bee colonies flying around. But the hives I put up were, of course, very, very conspicuous. The colonies, the wild colonies that I found, they're just flying in and out of a knothole in the top of a tree. And so I think so they're not so vulnerable to the bear. So we're not completely sure why they like the high entrances, but I bet protection, historically, evolutionarily, over evolutionary time, it was safer for colonies to have their entrance high off the ground. It may be better in the winter as well. It's a warmer, sunnier place. But I think the main reason is it gives them greater safety from big predators like bears.
So the question again is about urban beekeeping, and that if you have established some colonies that are hygienic, if they mate with drones from other colonies, presumably you lose that benefit very quickly.
Yes, undoubtedly.
Okay, we have one right at the back. So Roger has over the years, many colonies from buildings and other places where they're living. And he observes that the spacing between the combs tends to be sort of 38 millimeters and so on, wider than are normally used in commercial hives or whatever. What do you think that the benefit of a wider cone spacing might be to the bees?
Yeah, I don't really know, Roger. And this is Roger Patterson. Nice to hear from you, Roger. Yes.
Yeah.
And boy, that's a very interesting observation. And it makes me regret, Roger, that I didn't measure that when I was looking at the bee tree the nests in bee tree. You've got me there, both in terms of data and thoughts. Good question. Points out just how little there are many, many, many mysteries about the lives of these bees, for sure.
Yeah, right. Well, we're run out of our allotted time, so I think we'll have to let Tom escape. So, Tom, I just want to say thank you very much.
Thank you very much. It's my pleasure to join your meeting.